Dear listener, today we invite you to listen in on a recent broadcast we shared on LinkedIn.
And during that broadcast, we discussed tackling challenges to get advanced therapies to patients. And I spoke with Emmanuel Abate, our president of Genomic Medicine at Cytiva, and the CEO and co-founder of Replicate Bioscience, Dr. Nathaniel Wang.
So let's get down to advanced therapies. What's next? Which is really what matters on today's episode of Discovery Matters.
Our experts, we're going to talk about a very hot topic here at Bio, which is advanced therapies. Emmanuel Abate, 5 approvals on gene therapies just in the past year. Most recently, another mRNA therapy approved for the RSV illness. So much momentum. What are the problems we have yet to solve as an industry?
Science continues to progress,
right?
Okay, and evidence are there about the positive impact they have on healthcare and patients. But as far as Cytiva is concerned, we still consider we have a huge mission, an opportunity to improve the way those therapies are manufactured, so that they become more accessible, that we can scale up more easily.
The innovation doesn't stop.
The innovation doesn't stop.
We have been joined by Dr. Nathaniel Wang. You are the CEO, co-founder of Replicate Bioscience. Welcome. Replicate specializes in self-replicating mRNA. Tell us about that and some of your recent milestones.
Replicate specializes in self-replicating RNA technology, appropriately enough, for the development of different medicines. So what differentiates our technology is we've really worked on the vectors themselves. And those improvements really help the clinical bioactivity of what you're able to get with vaccines or different types of therapeutic expression of proteins. Another major differentiator is on the manufacturing front. With these longer RNAs, have really improved the quality and allowed for a much better safety profile that we were able to confirm clinically. We were able to get very good bioactivity for our rabies vaccine, all the way down to 0.1 micrograms, the lowest dose that people have ever seen in humans. So that's 1,000-fold less than what we saw with some of the COVID-19 vaccines for RNA. It also had very good durability. It actually matched the inactivated viral vaccine, which is currently commercialized. So the first demonstration of an RNA technology that can now match conventional RNA technologies. Very good safety profile, again, linked to some of the great manufacturing that we were able to do together. No severe adverse events across any of the doses tested. So,
but you have some, I'm sorry to interject, you have some milestones yet to cross, right?
Yes, absolutely. So we have some additional products that are being lined up to go into the clinic next year. So our Epstein-Barr virus vaccine that we're using your technology on as well as some other exciting projects that are in the hopper. So lots to come still clinically.
So for patients, what this means is what?
It's a great question. So once you have a technology where you've increased the therapeutic index, you're able to open up a few different things. So things where you target multiple proteins or pathogens, like combo respiratory vaccines, like flu COVID or flu COVID RSV combos as an example. Pediatric vaccines, where you need that good safety profile and that great bioactivity. So things like a prophylactic Epstein-Barr virus vaccine, and next generation vaccines for things that already exist. So I can improve shingles vaccine, where you can improve on the safety profile out there. So it really opens up a lot of avenues, and that's just vaccines.
That's exciting.
So I think, you both were talking about the momentum in the space from before. You can really feel it starting to build, and how these technologies are really enabling a lot of these advanced therapies. So we're very excited about it.
Super. What do you think the industry needs to do better so that innovators like Replicate Bioscience and other biotech companies can get to commercial success faster?
So I think the first thing that needs to be done is just generate more clinical data. There are all these exciting new technologies that are being developed, but until you can test them in the clinic and get a good sense on how they're performing, it can be difficult to know how to develop them as quickly as they should be developed. So that's the first step. The second is just building up the infrastructure. building up the raw materials that are needed to be able to advance these therapeutics, because the promise of nucleic acid technology is being able to go through drug development faster, but that requires the infrastructure to do it. And so as a field, we need to be very collaborative as we do it, because a rising tide raises all boats. And so it's important to find good partners and be very collaborative about it.
Indeed. Cytiva does a resilience index biannually, and one of the key findings is that 1/3 of the 1,200 industry executives who are interviewed do not feel that their countries are ready for cell and gene innovations in manufacturing. So there is a lot of progress yet to be made.
Absolutely.
Even with the momentum. Emmanuel, what are you excited about in the near future coming from the industry?
I'm in general really excited by the pace of science and the progress that have been made the past 5-10 years. I mean, I think, the work that Nathaniel and his team do, who would have thought 10 years ago that we would be where we are with mRNA, the work that is happening in cell therapy, in gene therapy, but also I'm thinking in what we've now sometimes called the more traditional biologics, but bispecific, ADC, radioligands. I mean, there is so much coming together. And I think, I'm finding very interesting that some of the biotech and pharmas are starting to look at multimodality approaches to single disease. I think the convergence of science around specific disease and pathologies is extremely promising for patients.
You're nodding eagerly, Nathaniel. What are you excited about in the near future?
I think he absolutely hit the nail on the head. And I share a lot of the enthusiasm about the momentum that the space has really been building. And I think what's been interesting is we saw those initial successes with emergency infectious disease vaccines. As you mentioned, exciting news about the RSV vaccine approval.
Yes.
And, a lot of the early work has been done on the more seasonal type of vaccine variety or the more emergency vaccines. So as we build up more clinical evidence and bring in new technologies, how can we extend on that? How can we tackle complex infectious disease? How can we go into some of these other therapeutic areas like therapeutic protein expression? And what are the technologies going to be that really enable us to make those jumps forward? So really excited for what's in the future there.
Do you see any threat to biology and the science with the advent of AI, or is that all benefits? Let's talk about another hot topic here, which is artificial intelligence. Where is that going? What are your comfort levels with it, Emmanuel?
I mean, in my mind, there is no doubt that there is huge opportunity in AI, right? I mean, certainly we're starting to use machine learning and complex modeling to improve some of our manufacturing processes. That's one piece, and I think we're only at the beginning. Design of media, design of lipids. I mean, all those things that are so heavy and labor intensive. I think AI could help, and of course, in drug discovery, I think we still need to frankly work on the use case, okay,
right,
and bring those use case forward, but. There is a huge momentum, and out of like 100 trials, maybe 20 are going to deliver something, right? So I think the game is still being played right now.
Yeah, it could be another one of those enormously time-saving, human energy saving, so that humans can devote energy to other pursuits, to something that is answering a more scientific question, perhaps. What do you think, Nathaniel?
I think from my perspective, the analogy that I would use is the initial sequencing of the human genome.
Yes.
And a lot of the genomics revolution that came. And, undoubtedly, it absolutely transformed the industry, and it created a lot of change. And with change comes opportunities, comes threats. So I think both of those things would be fair. And I think that I also agree that, it's about finding the right use case and how to integrate all of that information in those new tools. And I think that it makes for an exciting time. Absolutely. So I think it's going to bring a lot of positive changes.
Terms like mRNA become household words. Lateral flow is something we have available to us in our everyday vocabulary. What do you foresee when it comes to the general public's interest in this kind of technology and self-replicating RNA? Do you expect that to become a kitchen table topic?
Well, I would hope that it becomes a kitchen table topic, but I also hope that it becomes
softball question for you, Nathaniel.
Well, It's a complicated question. I think it's incumbent on the industry and all of us working on nucleic acids to be better about scientific communication, to make sure that we have a lot of transparency in the developments and not just the strengths, but the potential weaknesses of technology.
Right.
I think that what you don't want to happen with new technologies is for words like synthetic or things to become scary household names. And so I think it's important that we tackle those as an industry so that we can have the general public really share the enthusiasm that we believe that it deserves.
While acknowledging some of the challenges and some of the hurdles, perhaps, and the dangers.
Yeah, of course.
Yes.
I think that people can get distrustful if they feel like there's a lack of authenticity and transparency about the development of new technologies. And so, again, I think it's our responsibility to the general public to be as open about all of the challenges as possible.
Interesting. I'm going to volley the question to Emmanuel. What do you think is going to be part of our everyday vocabulary in the near future?
In a way, I was going to say, mRNA, cell therapy, gene therapy. At the same time, look, at the end of the day, there's patients that are in distress, right? Okay.
Right.
And I think, during COVID, it was interesting to see mRNA and testing becoming like part of the discussion. So I think it's more about, cure, right? Is what can we cure today that we would not even have dreamt of curing or moderating 10 years ago, right? And I think that's coming.
If you were to dream big, Nathaniel, what do you think is possible in 10 to 20 years?
I think that you're going to see that a much larger proportion of the new medicines that are developed are going to be RNA based. So, I think it was an interview in The Economist where it was saying in about 10 years, they were looking for a third of new drugs approved to be RNA. I don't know that I share quite that enthusiasm that it'll be that big, that quick, but I absolutely think that there's going to be an enormous growth. So I think that we will be able to get there eventually, in the next 20 years. So absolutely.
Terrific. Thank you. Do you want to take a shot at that, Emmanuel?
I'm thinking oncology in particular. Okay, I think some terrible disease will be curable in 10 years, and we've seen some
Speaking
of them
of
already.
moonshot.
And we've seen some of them in blood cancer, and that's now a reality. Okay, and I think we're going to see that I want to hope that we'll see that in solid tumors as well, right?
Right. Let's talk about access, right? Because all of this innovation ultimately has to reach the patient. But there are, to your point, Nathaniel, big challenges. Not every patient has the opportunity to receive these innovative therapies. How can we as an industry work towards improving access?
Yeah, I love this question in particular. Access for patients and for people globally is something that's really important to us in general. And I think that that's part of the beauty of this technology. So when you're using conventional technologies like recombinant proteins, the manufacturing can be very bespoke. It can be very complicated. Oftentimes it can be difficult to scale. And so the time that it takes to actually get to patients can be much longer. And in some cases, it can become so complex that it's not really practical to be able to do that. Because of a fundamentally similar types of manufacturing process you use for nucleic acids, you're able to leverage that to be able to push molecules through development quicker and get them to patients quicker. And as you start to bring some of these new technologies online into the wider public, things like self-replicating RNAs, these have much lower doses. And so the manufacturing footprint becomes smaller. This helps drive down things like the cost of goods, and that really improves access for everybody. And so, I believe it's really important that whether it's from the manufacturing side, the new technology side, from the infrastructure side, all of these things are things that really improve access for patients and things that the industry is already making leaps and bounds for, which is really exciting.
And there's a geopolitical element. Not every country can have major manufacturing sites. And then you have that challenge as well.
Absolutely. And that's where I think that being able to develop instrumentation that's more modular, that makes it easier to drop in to more distributed manufacturing facilities or technologies that reduce the amount of product that you actually have to produce, makes it so that you can really bring it to some of these other geographies. So absolutely, I think all of these things go hand in hand, and the improvements that we're just discussing are things that are going to be globally distributable.
Interesting.
I think so mRNA is a fantastic example of an opportunity to reduce cost of manufacturing. But when I think about the other modalities, it's going to continue to be about innovation. Think about the progress that have been made in the monoclonal antibody space in the past 20 years. There's no reason we cannot do that for viral production. There's no reason we cannot do that for cell therapy. And it's automation, it's new biologics, new cell lines, and we know what needs to be done, right? And so I think there is the industry has shown that through innovation we can bring the manufacturing cost down significantly. And to your point about globalization, that's what Cytiva does every day, right? With our flex factory solutions and enabling countries and making it easier to deploy capacity, right? It's about cost locally, but it's about how do you make it easy to deploy.
Now it's time for Every Day is a School Day. Dodie, have you got something you've learned since the broadcast with Emmanuel?
I do. And we happen to be recording during summertime, and so my personal reading has ticked up, which I'm a little bit proud of. So here comes a humble brag. But I just finished a great book about the K2 disaster from 2008. It was called Buried in the Sky. And my favorite science website, science.org, actually has a story about Mount Everest. So that's my theme today. And I found out that there is a lab in on the flanks of Mount Everest at the altitude 50 meters, 5,050 meters.
So that's like a scientific lab on the mountain.
Correct. Where they're studying the effects of high altitude on humans. They are studying climate change, biodiversity.
Because we already know why it's difficult to get a good cup of tea at 5,000 meters, because the water doesn't boil at the right temperature.
Depending on what you can pack, though. But sadly, this lab is losing its funding. So it's gone from a staff of more than 10 to a staff of 1. So that's today's story.
So one scientist.
One scientist in this lab
that's
at the foot
be lonely
of
and
Mount
cold
Everest.
And really unfortunate.
Right. So that particular scientist, whose name is, and I hope I'm pronouncing this correctly, Kaji Bista, a Nepalese native, is hoping to renovate and keep that lab up and running. It's a absolutely amazing story, though, is the equipment in that lab. It's just fully outfitted and really interesting to do research on our planet at that level.
It's extraordinary. So it's science at the real extremes, sometimes so often
Yes.
has real applications to, those of us who live in the quote-unquote normal world. Well, listen, from incredible heights
Right.
and scaling the highest mountains to, digging into the deepest depths of time. When we think about viruses, we often think of them as either bugs that infect us and make us sick, or, as we heard in today's episode, as delivery systems for advanced therapies. But actually, 8% of our human DNA actually comes from ancient viruses that infected our ancestors hundreds of thousands and even millions of years ago. So there's this paper in Nature Communications last month called Integrating Human Endogenous Retroviruses into Transcriptome-Wide Association Studies Highlights Novel Risk Factors for Major Psychiatric Conditions. Get that. They looked at the expression of these really old viruses in nearly 800 brain samples and cross-referenced it with data on genetic studies of psychiatric disorders, schizophrenia, bipolar disorder, depression, and guess what they found? Well, it turns out that these viruses were linked to the genetic risk of developing these mental health conditions. So it's just the tip of the iceberg. A lot more research needed to understand the role and the impact that these ancient viral sequences in our own DNA have on brain function and disease. But it's just this most intriguing line of inquiry that highlights the unexpected ways that evolutionary deep past continues to shape the biology and our health today. I think that's kind of poetic, you know? Viruses that plagued our ancestors now hold clues to the future.
Incredible.
It's extraordinary. And just imagine what's happened to us as a population over the course of the pandemic. How much viral load have we incorporated into our DNA that we're going to pass on to following generations and so on? So, absolutely fascinating.
Our producer is Beth Armit-Brewster, editing, mixing, supervision by Banda Productions in Sweden, music from Epidemic Sound, and my name is Dodie Axelson.
And my name is Conor McKechnie. I'm drinking Earl Grey tea. Make sure you rate us on Spotify or whichever platform you use to listen to our podcast. We'll see you when we come back with another episode of Discovery Matters.
Thank you for listening.